Okay, Assalamu alaikum students. My name is Dr. ERUM. I am the professor and head of Department Physiology at FRPMC. My email address is drerumimran@gmail.com. Today is the first lecture of hemopoietic module, and that is the composition of blood in physiology. You won't find this topic as such in any of the physiology book because it is actually the summarized form of the lectures which you will be doing subsequently in other lectures. So it's the summarized form. You can read it from my lecture, or you can gain some knowledge from the internet. But the plasma proteins which we are going to study in this presentation, I will teach you the functional aspect of the plasma protein, the structural and molecular level of plasma protein you will read in the biochemistry. So you can find that topic in the biochemistry books, but it's not available in the physiology books. Now, what are the learning objectives? The learning objectives of this lecture is: define blood, describe the properties of blood, discuss the composition of blood, define hematocrit or peg cell volume and its normal value, discuss the difference between serum and plasma, explain different type of plasma proteins and their function, discuss the conditions which lead to low plasma protein concentrations and effect produced by it, and enumerate the functions of blood. So, how we define blood? Blood is actually a specialized connective tissue. Now, why it is a connective tissue? Because like other connective tissues of the body, which are derived from the mesoderm, blood is also mesodermal in origin. And secondly, it connects different tissues and organs of the body because it's constantly flowing. It's the part of extracellular fluid, means it's present outside the cells. So that's why it is a part of extracellular fluid. It's about 8% of the total body weight. It's a complex mixture of formed elements and a fluid portion. Means it has got formed elements and a fluid portion. We'll discuss what are formed elements and what is the fluid portion. It has an average volume of about 5 liters in females and about 5.5 liters in males. Now, this is due to differences in body size between males and females. Okay, let's discuss the properties of the blood. The first property is the color. The color of the blood is red. The arterial blood is more bright red in color as compared to venous blood, which is purplish red in color. Now, the brighter red color of the arterial blood is due to the presence of high levels of oxygen. Okay, then the volume. The average volume in males is about 5.5 liters and about 5 liters in females, and I told you that this difference is due to difference in the body size. The pH of the blood is bit alkaline and it's about 7.35 to 7.45. As you all know that the pH of 7 is neutral and less than 7 is acidic pH and more than 7 is the alkaline pH. So what is the pH of the blood? It's about 7.35 to 7.45. Now the viscosity. The blood is about 5 times more viscous than water. Means it's more resistant to flow as compared to water. This viscosity is provided by the large amount of RBCs, and to lesser extent the presence of plasma proteins also make the blood viscous, but it's mainly the RBCs. So if there is increase in the number of RBCs, the viscosity is going to increase, and if there is decrease in the number of RBCs, the viscosity is going to decrease. Next is the property is the specific gravity, or which is also known as relative density. Now, what is actually the specific gravity? It's the ratio of density of a substance to the density of any given reference substance. Okay, since here we are using blood, we are taking the specific gravity of blood, so we will compare it with the water. Both are liquids, so we compare it with the water. So how you calculate the specific gravity of blood? That you take the density of blood divided by the density of the water. If the densities of the two substances, that is the blood and water, is same, then what will be the specific gravity? It will be 1. Okay, the densities are equal, so the specific gravity will be 1. But here the specific gravity of the blood is not 1, it's more than 1, means 1.050 to 1.060, means the blood is more denser as compared to water. Now, why the blood is more denser? Again, it is due to the presence of large number of RBCs. So, due to the presence of RBCs, the specific gravity has increased in the blood. Okay. So, any condition which lead to increase in the red blood cells in the blood, the specific gravity will be more. And if the number of red blood cells are decreased, the specific gravity will be less than water. Okay. Okay, now let's discuss the blood composition. Blood is actually made up of cellular portion and the fluid portion. The cells of the blood, they are also known as formed elements, and the fluid portion is known as the plasma. So out of 5 liters of blood, about 2 liters is the cells or the formed elements, and 3 liters is the plasma. Okay, here you can see below is the cellular portion, this, and above is the plasma portion. This is plasma. So the cells, they are also known as formed elements. Why they are known as formed elements? Because they are enclosed in a plasma membrane and have a definite shape and structure. Okay. And which type of cells are present? There are 3 different types of cells are present in the blood. The first are the red blood cells, which are also known as erythrocytes. Actually, the word erythro means red. The number is 5.2 million per millimeter cube or per microliter in males, and about 4.7 million per millimeter cube or per microliter in females. Now here you can see that male has got more number of RBCs as compared to females. Second cells are the white blood cells, also named as leukocytes. The word leuko means white. So white blood cells. They are about in thousands. The normal value is 4,000 to 11,000 per millimeter cube or per microliter of blood. The third cells are the platelets or the thrombocytes. They are in lakhs. They are about 150,000 to 450,000 per millimeter cube. So the largest number is of the red blood cells, which is in millions, the white blood cells, which are in thousands, and platelets, they are in lakhs, 150 to 450,000 per millimeter cube or per microliter. You do have to memorize all these values, they are important. Okay. So in the last slide we discussed that there are what is the composition of the blood, that is composed of cells and the fluid portion, that is the plasma. So now how you can separate these components, how you can separate the plasma from the cells. So what you do is you take out the blood, you put the blood in the test tube, then you add the anticoagulant, the heparin or the potassium oxalate. They act as the anticoagulants, means when you add them, they prevent the blood from being clotted. So now your unclotted blood, you put this blood in the spinner or the centrifuge machine. Now what does this do? It spins the blood at a very high speed. It is just needed to separate the components according to the densities. Okay, now once you spin it at high speed, then what happened that after some time you will see that these components, they have been separated according to the densities. The most dense are the red blood cells. They settle at the bottom because they are heavier, they are densest particles. Okay, they are most dense. Then is the buffy coat, which is formed by the WBCs and platelets. They are less denser as compared to RBCs, so they are just above the RBCs. And then is the clear fluid portion, which is the least dense. It is on the top. So this is how you can separate different components of the blood by using a centrifuge. You can not the centrifuge is not required for the separation. It actually speed up the process of separation. It just speed up the process. You can do this without the centrifuge also. What you do, you take the test tube and put the blood in it, add the anticoagulant and leave it in the test tube stand for about a few hours. And after few hours, what you will see that all the RBCs, they settle at the bottom. Then WBCs and platelets, they form a buffy coat or the creamy white coat, which consists of RBCs and WBCs. They are less denser as compared to RBCs, and the fluid portion is this plasma. So you can separate even without the centrifuge. But if you want to speed up the process, you use the centrifuge machine to separate these components. So here is the more clear picture. You can see the more denser RBCs, they have settled, which is about 45%. Buffy coat is formed by the white blood cell and platelets, which is less than 1%, and plasma is 55%. So this is how you can see clearly the separated portion of the blood. Okay, now another term, that is the hematocrit or the PEC cell volume. Now what is hematocrit or the PEC cell volume? How you define it? It's actually the percentage of the total blood volume which is occupied by the blood cells. What is it? Percent of total blood volume occupied by cells. How much percentage of cells is present in the blood? Okay, this is nothing but it is known as the hematocrit or the PEC cell volume. So it's nothing, in simple language we can say that it's nothing but it's actually the percentage of the cellular portion. How much percent of the cells is present? For example, if you take, first let me tell you that normal value of hematocrit in males is about 45% and in females it's about 42%. So I told you what is hematocrit. Percentage of cells is known as hematocrit, percentage of cells. So here cells, which cells? The three cells, RBCs, WBCs and platelets, their percentage. So the percentage of the formed elements or the cellular portion is known as hematocrit. So here you can see that when we added a blood in the test tube, we add the anticoagulant, we let them stand in this or we put it in the centrifuge machine, and then we speed up the process by this centrifuge, and we saw that the RBCs and the RBCs, they settle at the bottom, they packed at the bottom, and then is the buffy coat, which is less than 1%, and then is the plasma. So this cellular portion, when expressed in percentage, this is known as hematocrit or the PEC cell volume. So since buffy coat is very less than 1%, so mainly it is the RBCs which is contributing in the hematocrit. So whenever there is a deficiency of RBCs, then the hematocrit is going to drop. But if there is a deficiency of this white blood cells or platelets, the hematocrit is not going to fall because its normal value is 45% and it is represented by the RBCs. Okay. And in females, since there are less number of RBCs, so its value is about 42%. Now what do you mean by 45% and 42%? Actually, that when you take 100 ml of blood, in 100 ml of blood, about 45% is the cellular portion, and if 45% is cellular portion, then 55% will be the plasma or the fluid portion. And in females, the cellular portion or the formed element is about 42%. So what will be the fluid portion? It will be the 58%. अगर 100 ml of blood लिया तो उसमें 45% cells हैं और 55% plasma है, ठीक है. And if you take 100 ml of blood in females, the 42% it will be the hematocrit or the percentage of RBCs, and other 58% will be the fluid portion. ठीक है, पूरा 100% हो गया. Now, what will happen if number of RBCs are more? Means in polycythemia, the number of RBCs are more. So what will happen to the hematocrit? It will be more. When you take 100 ml of blood, उसमें अब RBCs ज़्यादा हैं तो ज़्यादा आ जाएंगे, and it will form more than 45% of hematocrit in males and more than 42% of hematocrit in females, and plasma will drop. ठीक है? What will happen in anemia due to low RBC count? If there is low RBC count, what will happen? You will see that hematocrit is going to drop and the fluid portion is going to increase. So here you can see that RBCs, they packed at the bottom. That's why this is also known as packed cell volume. Okay, now let's see what happened to the hematocrit in dehydration. So what is actually the dehydration? It means the loss of water from the body. Now since plasma is also the part of extracellular fluid, there will also be a decrease in plasma volume during severe dehydration. Whenever there is severe dehydration, the plasma is also lost from the body. The most common cause of dehydration are diarrhea and vomiting. Okay, so I have not used the word bleeding or hemorrhage. ठीक है, I have used the word dehydration. There is a difference between dehydration and bleeding or hemorrhage. Hemorrhage या bleeding हो रही है तो उसमें cells भी निकल रहे हैं क्योंकि blood निकल रहा है पूरा. But in dehydration there is loss of fluid. The plasma portion जो है वो loss हो रहा है. Blood is not lost. So what happened in dehydration? The number of RBCs in the body, they are not decreased. Okay, the number remain the same, but they get concentrated. है ना? Low plasma volume में अब वो हैं. So what will happen to the RBCs? They will get concentrated, and we say this as hemoconcentration. So in dehydration there is hemoconcentration. Okay, and when you will count the RBC in this, okay, in this test tube, in a dehydrated person, you take the blood, you centrifuge it, and you let it stand for some time, and then you calculate the hematocrit. What will happen to the value of hematocrit? The RBC count भी increase है और hematocrit भी बढ़ जाएगा, ठीक है? But this increase is the relative increase in the hematocrit, ठीक है? Relative increase का क्या मतलब हुआ कि in proportion कि प्लाज्मा वॉल्यूम ड्रॉप हुआ, इसकी वजह से हेमाटोक्रिट और आरबीसी काउंट आपको ज्यादा मिल रहा है, बट एक्चुअली इन द बॉडी नंबर ऑफ द आरबीसीज इन द बॉडी इज नॉट इंक्रीजिंग, ठीक है? वो सिर्फ कंसंट्रेटेड हो गए हैं. इस वजह से यू विल गेट हाई आरबीसी काउंट इन डिहाइड्रेशन एंड यू विल गेट हाई हेमाटोक्रिट when you will measure it, but actually in the body it remains the same, ठीक है? आपके number of RBCs in the body will remain the same. So there is a relative increase in the RBCs or the relative increase in the hematocrit actually, है ना? hematocrit बढ़ गया. So naturally when there is more hematocrit, it means that more number of RBCs, they are packed at the bottom, okay? And how it is going to be corrected? When you give adequate amount of fluid, the hematocrit will get back to its normal value. Here you can see that hematocrit has increased to 70% in a severely dehydrated patient. Means he has got severe diarrhea or vomiting, which leads to relative increase in the hematocrit. Okay. Now, which condition will lead to absolute increase in hematocrit? वाकई में hematocrit बढ़ गया. That is polycythemia. क्यों उसमें क्यों बढ़ गया? क्योंकि polycythemia is the condition in which there are more number of RBCs in the body. Production बहुत ज़्यादा है RBCs की. So जिस वजह से क्या हो रहा है? You can see that high hematocrit present in the polycythemia. ठीक है? Absolute increase हो रहा है. This is relatively increase है due to low plasma volume. Anemia है due to less RBCs, less number of RBCs. अब यहाँ है ही number कम, तो there will be less hematocrit. And अगर hemorrhage हो रहा है या bleeding हो रही है, तो फिर hematocrit पे क्या होगा? वो भी कम हो जाएगा. So in anemia and in hemorrhage, you will see what in anemia due to decrease in the RBC count in the body, there will be less hematocrit. But in polycythemia and in dehydration, in both condition you will find high hematocrit. देख लें आप दोनों जगह बराबर है almost. ठीक है. But यहाँ पर क्या है? Absolute increase है. यहाँ relatively due to low plasma volume की वजह से increase है. इसका treatment is to replace the fluid. When you will replace the fluid, the blood will get back to its normal concentration and RBC count and the hematocrit will come back to its normal value. Now, what is the difference between the plasma and serum? Plasma you have seen, how you collect the plasma. You have seen that plasma is actually a cell-free part of the blood. No cells are present. Straw color fluid over here. This is plasma. How you collect it? That you added the anticoagulant. You prevent the blood from being clotted. You take the blood in the test tube. You keep it straight in the stand. You can speed up by the centrifuge. The heavier part, the cells will be separated from the fluid part on the top, the cells on the bottom, and that clear fluid portion is known as the plasma. So this is the plasma. You got the plasma. But how you get the serum? Serum is again a liquid part of the blood. But what happens when you take the blood in the test tube? You do not enter the anticoagulant. Means let the blood to be clotted. The blood gets clotted after some time, a clot will be formed at the bottom. And the fluid which is present after coagulation is known as the serum. It means that serum has no clotting factors. Where are the clotting factors then? They have been used up in the formation of blood clot. So does plasma has clotting factors? Yes, because they have not been used. Because you have added the anticoagulant. You have prevented the blood from being coagulated. So here, how you can, if you have two fluid, one is plasma and one is serum, they are present in the lab. Because in many laboratory tests, for some tests you use plasma, for others you use serum. So how you can differentiate whether the fluid is plasma or the serum? Their color is almost same. So you can look for the presence of clotting factors. If clotting factors are present, it means that is the plasma. If there are no clotting factors are present, it means the sample is serum. But it's impossible to check so many clotting factors. Which clotting factor particularly you check is only the presence of fibrinogen. So if fibrinogen is present, it means the sample is plasma. If no fibrinogen, it means the sample is serum. So if we can say that serum is equals to the plasma minus the fibrinogen, means there is no fibrinogen in the serum. Now look at the plasma. Let's discuss the plasma. We have discussed that blood has two main components, the cells. We'll do the cells subsequently in different chapters. Now here we'll concentrate on the plasma. We discussed that plasma is about 3 liters and plasma is a straw color fluid. We have discussed that how you can separate the cell and from the plasma, from the blood to get the plasma. So if we look at the composition of the plasma, now we are discussing only this circled portion, that is the plasma. It's mainly made up of water. 90% of the plasma is made up of water. It also consists of plasma proteins. Very important proteins are present in the plasma, which are 6 to 8%. Electrolytes, since it's ECF, so it's rich in sodium and chloride, which are about 1%. And other components which are present in the plasma, beside these three main, they are the nutrients like glucose, amino acids, fatty acids, hormones. There are list of hormones which is present in the plasma, waste material, urea, uric acid, creatinine, and important blood gases, carbon dioxide and oxygen. They are also transported in the blood in the plasma. Okay, so these are the components of the plasma. Now here is actually the summary that whole blood is about 8%. You can see out of this 8%, you can see plasma is 55% and formed element is 45%. Then the plasma is made up of mainly water. It has got plasma proteins. It has got other solutes. They discussed that electrolytes, nutrients, gases and vitamins, waste products. Proteins, we'll discuss further that which type of proteins are present in the plasma. And these are the formed elements. There are three types of formed elements: platelets, white blood cells and red blood cells. The most numerous are the RBCs and the least are the WBCs. Okay, now let's discuss plasma proteins, which are the major component of the plasma. There are different types of plasma proteins. So let's see. What are the general characteristics of plasma proteins? There are actually three types of plasma proteins: albumin, globulin and fibrinogen. What are the three types of plasma proteins? In plasma, we find some proteins which are albumin, globulin and fibrinogen. This globulin is again divided into alpha globulin, beta globulin and gamma globulin. Okay. There are three types: albumin, globulin, fibrinogen. Globulin is again divided into alpha, beta and gamma. They are all synthesized in the liver. Means all the plasma proteins, they are synthesized in the liver except gamma globulins. Globulin के एक टाइप थी ना gamma globulins, जिनको हम immunoglobulins भी कहते हैं या जिनको हम antibodies भी कहते हैं. ठीक है? The gamma globulins, the immunoglobulins or the antibodies, they are not synthesized in the liver. They are synthesized by the B lymphocytes. Okay. All the plasma proteins, they are not pure proteins, they are glycoproteins. And they have a characteristic half-life in the circulation, after which they are degraded and they are formed again. Now this is the brief summary of all the plasma proteins. Here we can see that the first plasma protein is the albumin. It is smallest, very सबसे छोटा है, but its percentage is highest. It's 60% of the total plasma protein, where it is synthesized in the liver, and its main function is to maintain the osmotic pressure. How we'll discuss further. Okay. Second is the globulin. Its percentage is about 36%. It's the most abundant. Then is the globulin, which is about 36%. The globulins, they are divided into alpha, beta and gamma globulins. The alpha and gamma globulins, they are synthesized in the liver, but the gamma globulins, they are synthesized in the they are synthesized by the B lymphocytes, which are present in lymphatic tissues. Okay, and which act as the antibody. They are also named as immunoglobulins. The fibrinogen. It is the third plasma protein. It has a highest molecular weight. The lowest weight was of albumin, then was the globulin, And then highest molecular weight is of fibrinogen. It's a larger molecule, but its concentration, its percentage is least, that is, it is about 4%. It is also synthesized by the liver and it helps in blood coagulation. These are the specific functions of these plasma proteins. Okay, and alpha and beta globulins, they help in the transport of substances. Since plasma is water, so they transport mainly which type of substances, which are lipid soluble, which cannot travel in the watery medium of the plasma. So they need some transport proteins. So this transport proteins, they are alpha globulins and beta globulins. Now let's discuss these plasma proteins individually. The first and the most abundant protein, about 60% in the plasma, is the albumin. Its normal concentration is about 3.5 to 5 grams per deciliter. What do you mean by grams per deciliter? Means in per deciliter means 100 ml. So in 100 ml, how much amount of albumin is present? About 3.5 to 5 grams in 100 ml of blood. Where it is synthesized? It is synthesized in the liver, as other proteins, plasma proteins. Its half-life is about 20 days, and its measurement is very useful in liver function test, because liver is the factory which is synthesizing this protein, albumin. So this will give you an idea if blood albumin level is low. So you can say that maybe the factory is abnormal, maybe he's having some liver disease. That's why his albumin concentration has been decreased. So it's a very important index for to assess the liver function. Now, what are the functions of albumin? As I told you, its main function is to maintain the osmotic pressure. Now, what is osmotic pressure? It is the pressure which is required to stop the process of osmosis. Osmosis, you have studied in the chapter of osmosis, in the transport chapter, that osmotic pressure is preventing the process of osmosis. Since the osmotic pressure is here, it is actually provided by the proteins. The osmotic pressure which is provided by the proteins particularly is known as colloid osmotic pressure or oncotic pressure. Got it? Its oncotic pressure or the colloid osmotic pressure is nothing but it is the osmotic pressure which is provided by the proteins. And this osmotic pressure is actually dependent on the number of particles, not on the size of particles. So albumin सबसे ज़्यादा है, तो सबसे ज़्यादा कौन सा प्रोटीन जो है वो ऑस्मोटिक प्रेशर को कंट्रीब्यूट करेगा? Albumin. Then is the globulin, and least contribution will be by the fibrinogen. Okay. Actually, these plasma proteins, they have some generalized function and they have some specific function. Okay. Generalized function सबका जो proteins हैं, that's why they have some similar function. But कुछ उनके specific function है. तो albumin का सबसे main function जो उसका specific function है, that it provides the colloidal osmotic pressure or the oncotic pressure. And the value of this oncotic pressure is about 25 millimeters of mercury. So it is holding the water molecules. It is preventing the water from osmosis. Okay. So here you can see in slide, that it is providing the oncotic pressure. It is holding, these are the albumin molecules in gray, you can see, and water molecules in blue. This is a capillary, blood capillary, and how the water molecules, they are actually held by the albumin molecules, and the albumin, it holds the water molecule and it does not let the water molecule go out of these capillaries. It is holding it. But here you can see there is less albumin. Any condition which leads to low albumin. Now, so water holding capacity has reduced. So now water is moving through the capillaries and it is collected in the interstitial fluid, causing edema. Okay, so this is how the low albumin cannot now prevent the process of osmosis, but here normal amount of albumin and it is preventing the process of osmosis. So it is providing what pressure? It is providing colloid osmotic pressure or the oncotic pressure of value of about normal on colloid osmotic pressure value is 25 millimeters of mercury. Now conditions which leads to low plasma proteins. अब ये प्लाज्मा एल्ब्यूमिन कम कैसे हुआ? ठीक है, हमने डिस्कस किया कि एल्ब्यूमिन का मेन रोल है इन प्रोवाइडिंग द कोलॉइड ऑस्मोटिक प्रेशर और द ऑनकोटिक प्रेशर जस्ट बिकॉज ऑफ इट्स highest concentration, because osmotic pressure depends on the number of particles, not on the size, although its size is smallest of all the three plasma proteins, but since its number is more, so it's contributing more in the colloid osmotic pressure. Now what may be the causes of low serum albumin? Right, where the albumin is synthesized? In the liver. So any disease of the liver, any chronic disease particularly, ठीक है, the liver is chronically damaged, so हम कहते हैं उसको chronic liver diseases. So whenever there are chronic liver diseases, the albumin concentration will be low and patient come to you with the presenting complaint of edema. Okay, because fluid is being lost in the interstitial spaces. What may be other cause? Either there may be, or we can divide it into two causes. Either there is decreased production or increased loss of albumin. या तो production कम हो रही है या loss ज़्यादा हो रहा है. अब production कम क्यों हो रही है? Number one, due to liver disease, due to starvation and due to malnutrition. So there are three main causes of decreased production. या तो liver factory खराब है, या आप डाइट में प्रोटीन्स ही नहीं ले रहे हो, तो हाउ दीज प्रोटीन्स दे आर गोइंग टू बी सिंथेसाइज्ड, और इफ देयर इज malnutrition, ठीक है, आप खा ही नहीं रहे हो या आप डाइट में प्रोटीन ही नहीं ले रहे हो, तो व्हाट विल हैपन? द एल्ब्यूमिन लेवल विल डिक्रीज एंड पेशेंट कैन present with edema. Here you can see the picture of edema, that how this leg is swelled, and when you press it, a pit is formed, and that's why this edema is known as the pitting type of edema. Now, second cause, I told you that this albumin is being lost from the body. How this albumin has been synthesized, but it does not remain in the body, it is being lost in the body. It is being lost from the body. How it is being lost? The kidney disease. Normally, the kidneys, they does not filter the proteins. Okay, but if the kidneys are damaged, what will happen? The proteins, they are leaked. The lowest protein is the albumin. The lowest size protein is the albumin, so it leaks in the urine. So the albumin concentration will drop. Second is gut losing enteropathies. It means that your gut is abnormal, it cannot absorb the proteins, so proteins are being lost in the feces. And third cause of loss of albumin or the loss of protein is extensive burn, and the proteins, they are lost from the body through the skin. So all these conditions leads to low albumin concentration and leading to a clinical condition which is known as edema. Now, the second function of the albumin, that it helps in the transport of many different molecules in the plasma. Transport. The transport function was mainly performed by globulins. We saw that, है ना? So the main function, मैंने सबसे पहले बता दिया, albumin का the main function is to maintain the plasma colloidal osmotic pressure. But since it's a protein present in the plasma, it also helps in the transport of fat soluble substances in the plasma, which cannot be transported in the watery medium. The the plasma is all water, so the water soluble molecules can easily travel, but the lipid soluble molecules, they need to bind with some proteins, and those are the plasma proteins. But which plasma protein mainly transport? That is the globulin. But it also binds with some substances, like it binds metal ions, it binds free fatty acids, it also binds bilirubin, bile acids, and hormones. Mainly which hormones? Which are lipid soluble hormones. As you know, thyroid hormone and all the steroid hormones, they are lipid soluble, so they are carried by the plasma proteins. Okay, and they are not specific. Albumin transport proteins, they are not specific. You will understand this when you will study globulin. Other functions in general of all the plasma proteins is buffer action. This is performed by every plasma proteins, that they act as a buffer. Now, how they act as a buffer? Actually, the plasma proteins, they actually accept, they act as a weak base in the body, and they actually binds the H ions. They accept the H ions. So what will happen? So they remove excess H ions from the blood, as they are basic in nature. Okay, so they help in keeping the blood pH normal. The The other function of plasma proteins is it maintains the blood viscosity. You did in the first two slides, where you were studying the properties, that the blood is more viscous than water, 5 times more viscous than water, and viscosity is provided by the RBCs and to some extent by the plasma proteins. So it also helps in the maintaining the blood viscosity. It also acts as a reservoir proteins in starvation. So when a person is starving, is not taking his proteins, so these proteins can be used as a reserve proteins. Now the globulins. We discussed that globulins, there are 3 types: alpha globulins, beta globulins, and gamma globulins. Okay, there is further classification that alpha globulins, they are further divided into alpha 1 and alpha 2, then beta, and then gamma. Actually, this is the topic of biochemistry, and you will study these globulins in detail in the biochemistry. I'll give you some brief knowledge about this, the functions of the globulin. In general, all the globulins, they are mainly responsible for the transport of lipid soluble substances in the plasma, but they are highly specific to which they bind. To which substance they bind, it's highly specific. ठीक है? किस तरह से? For example, there is a thyroid binding globulin which will only bind the thyroid hormones. There is iron binding globulin which is known as transferrin, and which binds iron only. Okay, there are sex hormone binding globulin which binds only the sex hormones. So this is how they are specific to which substance they bind. ठीक है, अब थायराइड बाइंडिंग ग्लोबुलिन जो है वो आपका ईस्ट्रोजन को बाइंड नहीं कर लेगा, वो आपका आयरन को नहीं बाइंड करेगा. ठीक है, वो सिर्फ थायराइड हार्मोन को ही बाइंड करेगा. But this was not in the case with the albumin. They were not specific name. Albumin का जो मैंने बताया था कि transport is not specific. They can bind any substance in the plasma, but any lipid soluble substance. But these globulins, they bind the specific substance. Okay. The first function हो गया इसका transport, which is the main function. Will it contribute in the plasma colloid osmotic pressure? Yes, but to lesser extent, because its concentration, its number is less as compared to albumin. ठीक है, तो अगर आपसे पूछा जाए what is the function of albumin, तो आप उसमें transport नहीं लिखेंगे पहले. The main function will be to maintain the plasma colloidal osmotic pressure. But if someone asks you what is the main function of globulin, तो आप osmotic pressure नहीं लिखेंगे, आप क्या लिखेंगे? transport of substances, lipid soluble substances. Okay. Second, others, there are many different type of globulins in the blood, in the plasma, but you will study those in the subject of biochemistry. Here you must understand their help in the binding of lipid soluble substance. The blood clotting factors, जितने भी हमारे बॉडी में जो clotting factors हैं, they are either alpha और beta globulin. ठीक है, तो clotting में भी इसका रोल हो गया globulins का. And the gamma globulins, they help in defense function since they are the antibodies and they are produced by the B lymphocytes which are converted to plasma cells. Actually, the B lymphocyte, they forms the plasma cells and plasma cells, they form the antibodies. So defensive function भी होगी gamma globulins का खाली, ठीक है, और they are not synthesized in the liver, but all these globulins, alpha and beta globulins, they are synthesized in the liver. This is gamma globulins or immunoglobulin. This is another name for the antibody. These are again all the plasma proteins. They are glycoproteins. They are produced by the plasma cells in response to any antigen when comes in the body. The B lymphocytes get activated. They form the plasma cells and then they form the antibodies and they function as antibodies. The last plasma protein, which is of highest molecular weight, but it is less in concentration. It's also a plasma protein. It's also a glycoprotein. It's synthesized in the liver. It has highest molecular weight. And what is its role? Its role is in blood coagulation. And during the process of coagulation, it is converted into fibrin. And it forms a meshwork of fibrin, which helps the blood to clot. Now let's discuss the functions of the blood. The first function, we have given some main headings and we'll discuss the functions accordingly. The first function is actually the respiratory function. Now what is the respiratory function of the blood? That it transports oxygen from where? From lungs to tissues, and carbon dioxide from tissues to lungs to eliminate it out. Okay, so this is how it actually helps in the respiratory function. Other is the nutritive function. It transports the food from gut to tissues, so to utilize that food. So this is a very important function for the absorption. The food is being absorbed from the gut and is carried to different parts of the body. This is the function of the blood. Other is the excretory function. What excretory function? That it transports the waste material from which is derived during different metabolic activities. So the waste material is taken from the tissues and delivered to the kidney to be excreted out in the form of urine. These are urea, uric acid, creatinine, excess of water, all is excreted out through the kidney, but it is transported by the blood. Regulatory function. It regulates many things. One thing we have discussed, that all the plasma proteins, they regulate the pH because they act as basic and they accept the H ion C. Okay. So not only the plasma proteins in the blood acting as a buffer, but the hemoglobin and the buffers which are present in the blood, they are also maintaining the pH. But we are talking overall blood functions. So blood में कौन सी चीज regulate करती है? pH, plasma proteins, hemoglobin, since it's also a protein, and blood में there are different type of buffers which you will study, that all helps in the regulation of pH. एक तो ये हो गया. Second, it regulates the water content of tissues. How it regulates the water content of tissues? अगर water वहाँ ज़्यादा है tissues में, तो osmosis हो जाएगा water का from tissues into the blood, और अगर वहाँ कम है तो blood से water चला जाएगा. So there is a continuous exchange of water through the blood vessels to the tissues. Exchange हो रहा है, osmosis हो रहा है. ठीक है, तो this is how it regulates the water content of tissues, it regulates the pH. Third, it regulates the body temperature. how it regulates the body temperature, that what happens that when during different metabolism, the heat is being generated. Partly this heat is being used to maintain the body temperature. ठीक है, body temperature आपका 37 डिग्री सेंटीग्रेड पे मेंटेन रहता है. So this heat is being continuously produced to maintain your body temperature. Now excess of heat is being distributed by the blood, and as the blood flows to the surface of the skin, excess amount of heat is eliminated to the environment. So this is how it is regulating the body temperature. Okay, that it distributes the heat towards the surface, towards the skin capillaries, and through the skin capillaries it loses out to the environment, and so it regulates the body temperature. Another function is storage function. It stores and it makes these substances and water available to the tissues in need. अभी भी डिस्कस के वाटर कैसे वहाँ से चला जाता है. दूसरा हमने बात की थी कि प्लाज्मा प्रोटीन्स ड्यूरिंग स्टार्वेशन दे आर यूज्ड अप, दे एक्ट एज अ रिजर्व प्रोटीन्स, सो इट stores and then it deliver it to the tissues in the time of need. Next function is the protective function. It has got antibodies, antitoxins, white blood cells. All these help in the defense function of the body. Coagulation, I told you, fibrinogen and other clotting factors, they have a main role in the coagulation. We'll discuss this in detail in the subsequent lectures. And transport of hormones and enzymes. Very important function, that it transports different substances in the body, because hormones and enzymes, because hormones, they are directly being secreted in the blood. So all this, that they transport the substances in the blood. Okay, now look at the questions. First question is, what is the name given to the portion of blood having no fibrinogen and clotting factors? So there are four options: formed elements, hematocrit, plasma, and serum. So check yourself. What is the answer? Okay, I tell you the answer. The correct answer is serum, because serum has no clotting factors or the fibrinogen. Okay. Question number 2. In a normal sample of blood, which of the following cells would be most abundant? Lymphocytes, neutrophils, platelets, or RBCs? Yes, answer yourself. Now I tell you the correct answer. It's red blood cells. I know you must be knowing it. They are the most abundant cells, and these both lymphocytes and neutrophils, they are actually the types of WBCs, white blood cells. We will study them in detail afterwards. Okay, next question. Question number 3. Which plasma protein plays the largest role in maintaining the oncotic pressure of blood? You know, different plasma proteins. These are albumin, fibrinogen, alpha globulin, and beta globulin. So, which one is the right answer? Now I tell you the answer. The correct answer is albumin. Why albumin? Because it is in largest concentration in the blood. This plasma protein is about 60% in the blood. So that's why it is responsible for the, it has a major contribution in maintaining the oncotic pressure of the blood. Okay, thank you students. Thanks for listening, and any queries you can contact me via email. Wish you best of luck.